Good evening to all the NEET Aspirants 2026. Here we are in this session to discuss about biomolecules, carbohydrates. So far we have completed two chapters from cell biology, that is cell the unit of life and cell cycle and cell division. The most important concepts from those chapters are completed beta from our 100 concepts of NEET 2026 series. And here we are starting the third chapter of cell biology, that is biomolecules. Okay. So, today we are going to discuss carbohydrates from that. Before that, I would like to give you an introduction on NEET World's special programs. Those include like NEET World's 2-step to medicine for the students who are in 11th standard currently, who are aiming for NEET 2026. You can enroll for this course just at 3333 per month with complete study material both for online and offline students. Next, for 1-step to medicine, the students of 12th standard with the minimal fee again of 3333, and again for long term students you land on the NEET World's platform of Power Step to Medicine, where you will get again the complete course. Per month fee is just 3333. Okay. So, in this course itself your test series, your materials, your lectures, everything will be covered beta, and this is the minimal fee which is affordable for all the students of different financial backgrounds. To approach for these programs, you can call on to the given number, or you can visit our branch offline which is in Narayanguda, Hyderabad, of this address. And the new batch is commencing here from July 1st to 2026. Clear, with all these facilities, offline and online mode are provided. And now biomolecules, where we are going to deal with carbohydrates. Okay. So, here in biomolecules of carbohydrates, let us first look into what are the biomolecules. In the beginning of this chapter, if we have to see, the living tissue has been slurred with acetic acid, trichloroacetic acid, and then it has been taken into the test tube, wherein in the test tube after centrifugation you have got both acid soluble pool and acid insoluble pool. Acid soluble pool, which is the filtrate, okay, acid soluble pool, which is the filtrate. In this, all the inorganic molecules and those molecules which are of just 18 to 800 Daltons, 18 to 800 Daltons are kept in what? Are present in acid soluble pool. Fine. In this acid soluble pool, you will find those molecules which are inorganic, which have just molecular weight of 18 to 800 Daltons, and you can see biomicromolecules. Micromolecules. Fine. And then you have biomacromolecules, which fell in acid insoluble pool. Okay. Acid insoluble pool. So, this acid insoluble pool will be having all biomacromolecules. Biomacromolecules include lipids, nucleic acids, carbohydrates, proteins. Each day we will exercise one biomacromolecule. Definitely from these biomacromolecules you will get one question in your final NEET examination. So, here if you see beta, from all these biomacromolecules, lipids are not true biomacromolecules. Okay, that is because its molecular weight will be less than 800 Daltons itself. Okay, the molecular weight of the lipids will be less than 800 Daltons itself. Okay, but then these lipids are placed in acid insoluble pool because when all the living tissue gets slurred with trichloroacetic acid, what happens is the cell membrane will get ruptured, and cell membrane, you all know, it is 42% composed of what? Lipids only. Okay. So, these lipids will turn and form out to be vesicles. These vesicles are not soluble, that is why they have entered into what? Acid insoluble pool. So, if you have to see with respect to molecular weight, they will land in what? Acid soluble pool. But if you have to see with respect to solubility, they will land in where? Acid insoluble pool. This is the story of what? acid soluble and acid insoluble pools. Okay, and this is the story of biomicromolecules and biomacromolecules, where you will see in the introduction of this chapter. Now, coming to carbohydrates. What are carbohydrates according to you? You say, Kunal. What are carbohydrates?
Organic compounds.
Organic compounds. What are they composed of mostly?
Carbon, hydrogen, oxygen.
Yes, they are primarily composed of carbon, hydrogen, oxygen, CHO. Okay, they are H2N plus O, that's it, CN, H2N and oxygen. Fine. So, this is the general formula, and it is composed of what? Carbon, hydrogen and oxygen. Based on their composition, they are again classified into monosaccharides, derived monosaccharides, oligosaccharides and polysaccharides. Before that, you know one thing, that all carbohydrates are otherwise called as saccharides. Okay, carbohydrates are otherwise called as saccharides because their basic component is what? Sugar. Fine. So, what is the synonym of carbohydrates? Saccharides, because their basic component is sugar. Okay. As such, you will be having two types. It can be small sugars or large or complex sugars. If you have to see biomicromolecules based on the number of carbon atoms present in them, okay, and how far they can be simplified more. Based on that, if you have to see beta, the smallest carbohydrates are what? Monosaccharides. And then derived monosaccharides, oligosaccharides and polysaccharides. Let us look into each type of carbohydrate. First is monosaccharides. Monosaccharides are the simplest form of sugar beta. Hardly 3 to 7 carbon atoms will be present. Okay, it is composed of 3 to 7 carbon atoms. It cannot be further hydrolyzed or simplified more. Okay, these are the simplest form of carbohydrates. They cannot be hydrolyzed more, and these are biomicromolecules only. Okay, best examples of monosaccharides includes glucose, fructose, mannose, galactose and hexose. And all these monosaccharides are said to be reducing sugars. Reducing sugars. What are reducing sugars?
Which oxidize themselves and reduce others.
Yes, very good. And one more point to be remembered here beta, they have free aldehyde and ketone groups. Okay, can you see here in glucose that free aldehyde group, in fructose free aldehyde group, in galactose free aldehyde group. So, this free aldehyde group will react and reduce the other components. Like this are said to be reducing sugars, and they are identified by Felling's test, that test which is used for urine test. Okay, Felling's test, where this will reduce cupric ions to cuprous ions. Fine. After adding Felling's reagent to this will convert cupric ions to cuprous ions, where this is reducing the copper. So, by this we can say these are reducing sugars. They will be having free aldehyde or ketone group. Clear? So, this is about monosaccharides introduction, and these are the examples you have to remember. Some key points that you have to remember with respect to glucose, fructose, I will tell you. See, glucose is also known as dextrose, or it is simply called as grape sugar or corn sugar, and it is universal sugar. Glucose is called as universal sugar, and fructose is the most sweetest sugar which is naturally available. Okay, so this is known as fruit sugars, or this is also said to be levulose. And monosaccharides will have free aldehyde and ketone group, that is why they are called as reducing sugars. Clear, right? Clear everything with monosaccharides?
Glucose is primary energy source.
Yes, that is the primary energy source in plants. Okay, animals too. Okay, next, saying you about the derived monosaccharides. Derived monosaccharides. Tell me a few examples of derived monosaccharides. Of course, it is not there in your NCERT, but then this is there. Ribose and deoxyribose structures are there, right beta? Ribose and deoxyribose. ribose and deoxyribose are kept under derived monosaccharides because they are little modified monosaccharides. They are also the simplest sugars, but they are what? Little modified monosaccharides. Fine. First let us look into the ribose structure. Ribose or deoxyribose is how many carbon? 5 carbon. So you can call it as pentose sugar, right? So from here we will start, right? Usually, so as such you will be having 5 carbons, okay? In the second carbon, this OH and O that you already know, but directly I will draw out, I will come to the second carbon because second carbon will show the variation in deoxyribose and ribose sugar. In deoxyribose, if you have to see beta, you can see OH group at second carbon, right, and H here. This OH group, presence of OH group at second carbon will make it this pentose sugar what? Ribose. Same the case, everything, the structure will be same for deoxyribose also, but at the second carbon oxygen is absent. Okay, at the second carbon oxygen is absent, so it has only H. Okay, that is why it is called as deoxyribose sugar. Fine, fine. This is deoxyribose and this is ribose sugar. Both of them are pentose sugars only. These are little modified monosaccharides. Okay, this you will see where? In the nucleotides of DNA. In the DNA, which is a polynucleotide chain, there in each nucleotide the sugar molecule which is present is what? Deoxyribose. In RNA you will find ribose sugar, that is it. Okay. Next, if you have to take one more example like glucosamine, that is also derived monosaccharide, okay, which has amino group over the glucose as such. Next, oligosaccharides. These are also the smallest carbohydrates, but then they are formed by condensation of 2 to 9 monosaccharides. How many monosaccharides ma? 2 to 9 monosaccharides are condensed forming what? The oligosaccharides, which are small carbohydrates. Monosaccharide units, they are joined by what? Glycosidic bonds. I will say you, we have 1,4 glycosidic bonds, alpha 1,4, beta 1,4, alpha 1,2 glycosidic bonds. We look into all the oligosaccharides now. So these glycosidic bonds are usually bound at 1 and 4 carbons of the neighboring sugar units, neighboring monosaccharides you can say. And most of the oligosaccharides or polysaccharides, they will not be reducing because the aldehyde group or ketone group whichever is present na, that will help in binding the preceding sugars, neighboring sugars, okay, that are involved in what bond? Glycosidic bond. So they are not free. When there is no free aldehyde or ketone group, no reducing agent. So here if you see ma, based on the number of monosaccharide units, you will be having disaccharides and trisaccharides. Disaccharides best examples include sucrose, maltose, lactose, trehalose. Trehalose you will find in the insects, okay. And trisaccharides, if you see, examples include raffinose, and they are made up of glucose, fructose and galactose. Clear? Trisaccharide, here you can see the combination of 3 monosaccharides, okay. So here you see ma, glucose. 2 glucose units will combine to form what? Maltose, which is a disaccharide. So here you see carbon 1, 2, 3, 4 and this is 5th carbon. Same case here, C1, but from here we have to count, right, because glucose will be having 6 carbons, not 5 carbons. But you listen here, this is carbon 1 and this is carbon 4. This will be 5 then.
The one CH.
Yeah, this is 6. Yes. Now it has come. See. C3, C4, this is C5, this is C6. Fine. So 6 carbon compound. Here you see Nana. Carbon 1 of this glucose is attached to 4th carbon of this glucose. This is called as what? Glycosidic bond, wherein yes, you can assume like OH of this is combined to H of this, like that you can assume. Fine. This will lead to what? Dehydration. Okay, what's happening over here? Dehydration. Yes, H2O is being removed. Dehydration, which leads to alpha 1,4 glycosidic bond. Alpha 1,4 glycosidic bond. Just a second. Alpha 1,4 glycosidic bond. Fine. Next, here if you see lactose, lactose is also in between glucose and galactose. Okay, this disaccharide is in between what monosaccharides? Glucose and galactose. Both of them will have different structures. Okay, so here if you see C1, 2, 3, 4, 5 and here 6. Same the case 3, 4, 5 and 6. What connections are there? C1 and C4. Again alpha 1, but here we call it as beta 1,4 glycosidic bond. Okay, this is called as beta 1,4 glycosidic bond. This is alpha 1,4 glycosidic bond in maltose. This is in between two different sugars, lactose, galactose and glucose. Remember that. In sucrose if you take it is in between glucose and fructose. Again if you have to see what carbon is binding, C4, C5 and C6. Here also you see C5, 3, 4, fine. So here also if you can observe it is C1, C4, okay. That is also 1,4 glycosidic bond. Clear, right ma? So here you can observe only one thing, that is oligosaccharides. You have to remember the disaccharides, okay. Disaccharides which have two different monosaccharides. So maltose is a homosaccharide wherein it has two glucose unit, but this is hetero where it has glucose and galactose. If you see sucrose, it is also not homo, it is having glucose and fructose units. Clear, right? Next, polysaccharides. Polysaccharides are long chains of carbohydrates. These can be the threads containing different monosaccharides units. These monosaccharides are called as building blocks. Fine. And these are clearly seen in the acid insoluble pellet of polysaccharides. Polysaccharides will be of two types, homopolysaccharides and heteropolysaccharides. Here you can see the linkage of polysaccharides, that is through glycosidic bond. Just now we have seen that alpha 1,4 glycosidic bond, and that is through what? Dehydration or removal of water molecule. Yeah, you can see here the types of polysaccharides. Types of polysaccharides are formed based on what? Based on the composition of the sugars, okay, whether it is composed of same monosaccharides or it is composed of different monosaccharides. You can clearly see the definition here. They are formed by polymerization of only one monosaccharide monomer. Here they are formed by condensation of how many? Two or more monosaccharides or their derivatives. Best examples here are starch, chitin, glycogen, cellulose. Here peptidoglycan, agar. Clear? Here you see cellulose, okay. First example is cellulose. Cellulose is a homopolymer of glucose, and where do you observe cellulose more? Cell wall of what? Plant cell, okay. So this cellulose which is composed of glucose units are helically arranged in that. Yes, unbranched, helically arranged. Look into the theory once beta. So this cellulose is a polysaccharide. This contains only one type of monosaccharide. That one type of monosaccharide is what? Glucose. Okay. And how two glucose units are linked here? By beta 1,4 glycosidic bonds. Clear? So plant cell walls here are made up of what? Cellulose, where paper is made from plant pulp. Okay, paper is made from plant pulp, and cotton fiber, these are cellulosic. These are the direct lines that I have extracted from the NCERT. So pay keen attention and look into it. Okay, cellulose does not contain any complex helices. Okay, and it is hence this does not hold the iodine. Cellulose will not give blue color, that is because it cannot hold the iodine, because it does not have complex helices as that you see in starch. Fine beta, understood the points of cellulose? Next, Yeah, starch if you see ma, this starch is a homopolymer of alpha D glucose. Fine. And starch is composed of two types of polysaccharides. You call it as?
Amylose and amylopectin.
Very good. Amylose and amylopectin. Actually, this is a very branched polysaccharide. Cellulose is unbranched, but this is branched. Here in that unbranched units, you can see alpha 1,4 glycosidic bonds. That unbranched is what? Amylose. But at branched points, you can see amylopectin with what units? Alpha 1,6 glycosidic bond. Carbon 1 of one glucose unit and carbon 6 of another glucose unit will be connected. Any doubt?
D-glucose alpha 1,4.
Dextro and levo.
Third. Amylopectin is branched chain. D-glucose units linked by alpha.
Alpha 1,4 glycosidic bonds only, but you will see alpha 1,6 glycosidic bonds only at the branch points. Okay, else everywhere you will observe only alpha 1,4 glycosidic bonds. Don't get confused beta. Only at branched points you will see alpha 1,6. Clear na? Here you see 80% of starch is made up of what? Amylopectin only. Only 20% is made up of what? Amylose. And it has complex helices units because it has branches of amylose and amylopectin. As such, it will give what color with iodine test ma? Blue color in iodine. A blue color in starch test, we will call it as with iodine. Okay. So, that is why starch is what? Stored food material inside the plant cell. What is the stored food material inside the animal cell? Glycogen. Glycogen will be giving red color with iodine test. Okay, remember that. This will give blue and that will give red color. Next, glycogen if you see, polymer of alpha 1,6, alpha 1,4 glycosidic bonds and alpha 1,6 glycosidic bonds as well. Here in this, if you see this diagram which is in your NCERT, clearly stating the right end and the left end. Okay. So, the right end here you can observe ma, free aldehyde groups are present, right? So, here at the right end you can observe reducing sugars. Whereas at the left end, if you see in the left end, these are called as non-reducing end. Left end is called as what? Non-reducing end, because there are no free aldehyde or ketone groups. They are connected with alpha 1,4 or alpha 1,6 glycosidic linkages. So, this end is what? Non-reducing end. 1,4 and 1,6 also beta, because there are no free aldehyde and ketone groups, right? They are involved in connecting for the glycosidic linkages. Clear right now? And here you see lastly chitin. Where do you see chitin?
Skeletonization of fungus, porifera and
Many means arthropoda, can you see? Yeah, yes, arthropoda. So, chitin is found as the structural component of fungal walls and exoskeleton of arthropods. Direct line from your NCERT. Okay. And this is the basic unit of what? Chitin. What is the basic unit of chitin? NAG units we will say. Those are N-acetyl glucosamine. N-acetyl glucosamine. Fine, N-acetyl glucosamine. And this is an unbranched polysaccharide. These are again joined by what? Alpha 1,4 glycosidic bonds again. The monomers of N-acetyl glucosamine will be like glucose itself in chitin. Okay, this is homopolymer. Okay, so it has what? Glucose units only. That is why we will call it as, we call chitin as what is? Fungal cellulose. Yes, it is also called as fungal cellulose. Fine ma. Next, inulin. Inulin is nothing but it is a polymer of fructose. Again this is what? A homopolymer only. Homopolymer. Fine. This you will see in the stored food material in the root regions of one plant which starts with V. I will say you tomorrow, I will say this point tomorrow. So, this is all about carbohydrates. So, what we have discussed today? Carbohydrates are also called as what? Saccharides because they are what? Sugar units. And we have exercised the different types of carbohydrates. What are they? Monosaccharides,
Oligosaccharides,
yeah, derived monosaccharides, oligosaccharides, polysaccharides. Best examples of monosaccharides? Glucose, galactose, fructose. Okay, these will be the best examples for monosaccharides. Mannose also. Whereas maltose is a disaccharide with glucose units, okay, which are linked by what bond? Alpha 1,4 glycosidic linkages. Fine. Lactose is having what? Galactose and glucose with again 1,4 glycosidic bonds, and sucrose is having what? Glucose and fructose. And that comes under what?
Hetero.
Yes. They are not polysaccharides, they are oligosaccharides, right?
2 to 9 carbons.
Yes, 2 to 9 carbons. And how about derived monosaccharides? For example,
ribose and deoxyribose.
very good, ribose and deoxyribose. Those are what? Derived monosaccharides. Of those two, which will not have the oxygen group at second carbon? Deoxyribose. And do you see that deoxyribose in RNA nucleotide chain? No. And one more simple question. Yeah. Which is the most sweetest sugar?
Sucrose.
Like
Fructose.
fructose. Okay. These are simple points only, okay, but you should not forget that. Best examples for oligosaccharides, disaccharides and trisaccharides. For trisaccharide, raffinose is the best example. Combination of what? Glucose, fructose and galactose. And is cellulose a complex chain? Unbranched helical chain. Will that give blue color with iodine? No.
It cannot hold I2.
Yes, that's it. Starch will give what color with iodine? Blue. Glycogen?
Red.
Yes. That's it, right? Inulin is a polymer of what? Fructose. Chitin is also called as? Fungal cellulose. It is a polysaccharide of what? Glucose units. Okay. And in a glycogen chain, at what end do you find reducing sugars? Last question. Right end. Why? Because they have free aldehyde groups. Okay. Yes, that's all for today's class. Tomorrow we will deal with proteins. Okay. And we will exercise few questions as well. Thank you.